Welcome to Stock Therapy with Penny Queen. Today, I have CEO Fahar Merchant and CFO Liz Williams with Medicenna. And today we're going to cover the company, the changes they've had over the past couple of months, and then we're going to dive into some technical questions. So welcome both.
Thank you very much. Nice to be on your show again. Thank you.
All right. Well, first thing I want to do, because I know we'll have people watching that didn't watch the prior two videos. Can you cover exactly what Medicenna does?
Yes. So Medicenna really is a company that's focused on immuno-oncology primarily. And what we are doing is using evolutionary techniques to create better molecules. And that particular approach is being focused on what we call interleukins. So interleukins are essentially molecules that we all have in our bloodstream that are there to modulate or control our immune system. Now, that immune system can be used prominently to fight cancer. So what we are doing essentially at Medicenna is engineering these interleukins so that they are best able to stimulate the immune system to fight cancer, but at the same time be safe for the patient. And that's the focus. And around that focus, we have three different interleukins, interleukin 2, 4, and 13, each one of them that we are developing. And the one that is obviously the most advanced amongst the superkines is interleukin 2, which is the drug we call MDNA 11. And a second program, which is based on interleukin 4, which is being used to treat patients with recurrent glioblastoma. That particular program is known as MDNA 55. Both of these programs, both MDNA 11 and MDNA 55, has generated really exciting data to validate and confirm that the approach we are taking in terms of engineering these interleukins is very unique, very differentiated, and therefore really promising so far. So really excited with the data we have to date. Okay.
All right, perfect. Liz, can you touch on the cash you have on hand and what that will cover as far as future work?
Yeah, so our last reported was September 30th. So we had just over 40 million Canadian available to us at that point in time in cash and cash equivalents. And that takes us into the second quarter of calendar 2024. So the ability study is fully funded. That includes the dose escalation portion that we're currently dosing patients in, as well as the phase 2 monotherapy expansion portion of the study, which is an additional 40 patients, and the combination arm of the study, which is MDNA 11 plus Keytruda, and that as well as another 40 patients. So an additional 80 patients in the phase 2 portion of the study. We anticipate that'll be complete in terms of enrollment by the end of calendar 2023. So we're fully funded through those key milestones and data readouts.
Okay, beautiful. So that is the current MDNA 11 dose escalation, and then the monotherapy is much healthier patients, right, with just the MDNA 11.
Correct.
So, and then of course the Keytruda, both of those together. I think that's one of the things that that a lot of people aren't picking up on. I know with the last data was, 5 out of 14 of your patients were showing tumor control, but I think people aren't picking up that these are very sick patients. I know, I think like 79% had already failed immunotherapy, and a lot of these are late stage. So it'll be very interesting to see what MDNA 11 does on its own in healthier patients. So Liz, right now you're trading at just about cash on hand. I know you've fallen below $1 on the US side. What do you think the danger is of Nasdaq delisting?
Yeah, it's a good question. It's something we're watching very closely. Our first 6 month period ends April 24th, 2023. And based on where things are at now, we would qualify to get an additional 6-month extension. So we'll plan to do that. And I think, as Fahar mentioned, we will be kind of well past some very meaningful data readouts before we get to that next 6-month period. So our hope is that we'll be trading well above the dollar mark, and it'll be not an issue by the time we get to the end of our second extension. And depending, we will have an update in Q1 as well. So there's another opportunity there for the share price to lift and get past the $1 threshold. But we're monitoring it very closely, and we will apply for that second extension if we need to, for sure.
Perfect. Yeah. And I know there's only about another, month left of tax loss selling. And I know that all sectors have been being hit pretty hard, but definitely having those catalysts coming up between now and that April date is excellent. So can you talk about the upcoming catalysts?
Yes, so the key catalyst coming up, of course, is first and foremost to establish what we call the recommended phase 2 dose. So that generally occurs during dose escalation, which is currently underway. As you know, we are in cohort 5 now, where patients are receiving the 90 microgram dose. We might have a cohort 6 as well, but the key thing here is to establish the optimum dose, which is efficacious, but at the same time safe for patients, so that we can then start the dose expansion part of the study. So the key milestone, first and foremost, is completing the dose escalation phase. The second milestone, which we expect, and we'll have early readouts on those data, is the dose expansion, where MDNA 11 is being used on its own. Now, remember, this is phase 2 portion of the trial. And these patients will be, as you mentioned earlier, much more healthier. Typically, the patients that are currently in the phase 1 dose escalation phase would not be the patients who would generally qualify for the phase 2 portion of the trial, because in phase 2, we need healthier patients, not the type of patients we are currently seeing in phase 1. Okay. And then finally, towards the end of the year, we will have early data on the combination of Merck's Keytruda with our MDNA 11. So before the end of the year, that is next year, we'll have data that will share from at least 80 more patients, half of them receiving MDNA 11 on its own, and the other half receiving the combination. So lots of data coming up middle of next year and towards the end of next year as well. And then in the first quarter of this year, or rather next year, we will obviously have established the recommended phase 2 dose. Okay.
Okay. Perfect. And so I wanted to touch on a little bit, right now you're sitting at a market cap of about 30 million US. So I know there's been a lot of tax loss selling over the past month, but also there's been changes in the IL-2 space that I think a lot of investors might be misinterpreting. So I see it as positive changes for Medicenna. What are you thinking?
Well, it's on the one hand, if you really look at Medicenna, we have always believed that the way we've designed our molecule is very different from the others. So, just because the other programs, specifically, let me mention Nektar, that failed at the beginning of this year with their phase 3 trial. And then more recently, we've seen Sanofi go back to the drawing board with their version of the IL-2. And more recently, we've seen Neoleukin abandon the program. In each of these 3 cases, there were some serious concerns with respect to the design of the molecule. So we feel very confident that the approach we have taken clearly bypasses all those liabilities that those programs had in terms of drug design. So that's, on the one hand, important because clearly we had companies like Nektar and Syntorex that were 2 to 5 years ahead of us, and they are no longer ahead of us now. We are sort of getting to the top 2, if you may, in terms of companies that are advancing the IL-2 programs. So we are really positive with the expectations with the way our drug has been working, particularly in the phase 1 portion where the patients are really sick. So that's, from our perspective, a strong indication that we are on the right track. And I think if you look at some commonalities as to what happened in the other programs, with Nektar, with Sanofi's drug, both those drugs essentially use the same old proleukin drug. And we know that Proleukin has a number of liabilities, and therefore not surprising that those programs are not showing the kind of activity or did not show promise. So that's one thing. Second, all the three molecules that we're dealing with, all of them are pegylated. That means they have polyethylene glycol used to extend the half-life. And we know that polyethylene glycol is really not an sort of an ideal molecule to extend half-life. It has its own liabilities. So when you have liabilities of polyethylene glycol, and then you have liabilities of Proleukin, you combine those together and you really don't get the best of both worlds. You get the worst of both worlds. So we have avoided pegylation. We're using albumin, which is the most abundant protein in our bloodstream. And therefore, that's from that perspective safe. And the important thing is albumin actually accumulates in the lymphatic system. It accumulates in the tumor. It accumulates in the tumor draining lymph nodes. This is where all the immune cells are. This is where you need your drug to be present so that it can boost the immune system where it's actually needed, rather than have something such as pegylation, which does not accumulate anywhere. And therefore, you have those final issues. And I think one other thing that your listeners might be concerned about is Neoleukin. So Neoleukin was the most recent program that announced that they are not pursuing the IL-2 program, but their approach has been very different. They've used a very novel technique called computational methods to sort of design a molecule from scratch. So their IL-2 has no resemblance to any human IL-2. In fact, 85% of the molecule has no resemblance to any protein of the human protein sequence. So therefore, you're suddenly injecting something that you know very little about. And therefore, not surprisingly, they were not able to pursue the program further. In our case, what we've done is we've taken the Proleukin, we've inserted 7 key mutations that improve the efficacy of the drug and they improve the safety of the drug. So those two things are the key issues with Proleukin that we have eliminated from the design of our drug. So that's where we feel very confident about, and the sort of the early data we've seen already.
Now, if I can back to pegylation, now my understanding is that's done to make a larger molecule so that it doesn't get washed out. So
Right.
probably not the right terms, but that's antifreeze, isn't it?
It is,
Polyethylene glycol.
no, it is antifreeze. It's something
Okay.
used in
Just
shampoos.
checking.
No, you're right. Polyethylene glycol is the type they use is essentially based on the same core polyethylene glycol that you use in antifreeze and shampoos, etc. So it sort of makes the molecule much bigger. So what they use polyethylene glycol is by making the IL-2 bigger, it doesn't clear to the kidneys as fast or as easily. So it stays in the bloodstream much longer. Okay.
But the albumin,
it does the same thing. It makes the molecule bigger, but at least you're not using antifreeze. You're using albumin. So it's sort of and albumin has these unique characteristics. And that is the important thing, is it accumulates in your lymphatic system. That's where your immune cells are, right? So that's where you want to stimulate your immune system.
Well, I noticed that your patients were being given medication once every 2 weeks by IV. And that was it. So, I mean, that kind of tells you right there that we know it's staying in the system long enough. So,
yeah.
All right.
Yeah, not only that, I think what we found also is that even after we injected the drug, we found that the immune system was still activated even after 11 and 14 days. So that it's showing that our drug's effect is long lasting. And that can only explain by the fact that it's staying in the bloodstream longer, but more importantly, it's localizing in the lymphatic system in the tumor for much, much longer.
Okay. And before I get into the technical questions, I want to kind of go back to something we talked about last time, was that there's been all of this M&A activity in the space, but it's been happening. Some of it was, I think, like pre-IND. some of this is, it's been happening all over. So I just want to make sure that everybody realizes that the data you'll be reading out this year is really advanced by comparison, and that it's just been, such high quality. I am really excited to see healthier patient data. So, all right. So everybody's going to have to bear with me on these questions because I'm going to have to read a lot of them because these were sent in to me and a lot of them are pretty complicated, but fortunately this first one is not. So people want to know about the data that you're seeing now on MDNA 11 with the dose escalation phase. Can you compare that to the preclinical data?
Well, I mean, there's two sets of preclinical data, right? So the first one was what we did in mice, for instance. And of course, there we saw that the drug was able to really shrink tumors, the majority of different type of tumors, very efficiently. And it was able to do its job on its own. It did not, it certainly helped when you combine it with the checkpoint inhibitor, but we saw that the drug was active on its own. So what we are seeing now in the clinical data is something similar. We're saying, okay, these patients are really sick, unlike the mice that were sort of healthier mice, but here we've got very sick patients that generally would not qualify in phase 2 or phase 3 trials, or they've run out of all the other approved options. So to see that we are observing stable disease in 5 out of 14 patients, including a pancreatic cancer patient where the tumor continues to shrink, that's really encouraging and unexpected for us. We were not expecting this kind of data that soon. We were sort of expecting to see responses in patients once we had the next phase of the study, the phase 2 program started. So that's sort of an indication that, while it is clearly showing something that we saw in mice. And as far as the non-human primates or the monkey studies that we conducted, we are seeing very similar stimulation of our cancer-fighting immune cells, except that we are seeing the stimulation of cancer-fighting immune cells at much lower doses than what we saw in monkeys. So that also is a confirmation that our drug works even better in humans at much lower doses, and therefore is consistent with the fact that we know that monkey IL-2 receptors or monkey's immune system needs a much higher dose with IL-2 for it to be stimulated. So this is, again, very consistent with what we're seeing, that humans are responding to the drug at lower doses, and therefore that's consistent and encouraging that we don't have to go to the high doses that others would need to, right?
All right, perfect. This one was also submitted. It says, I understand the logic from safety perspective regarding the step-up dosing, but it's also known that IL-2 is most potent at the first dose. Is there a risk with step-up dosing compromising efficacy?
Good question, really good question. And I think one of the things is that we need to hit the tumor hard, and that's really important. And we know that even at 30 micrograms, the step-up dosing, that the dose we're using, we are stimulating the immune system very substantially. So we are getting 2 to 3-fold increase in lymphocyte counts in our cancer-fighting immune cells, the CD8 T cells, etc. So from that perspective, I think that's really important that we're already seeing, even in a patient that received just 10 micrograms per kilogram, we're seeing that patient with stable disease and now has been receiving the drug for over a year, and the tumor is really well controlled. So that's going to show us that although we are using what we call a step-up dosing, the 30 microgram dose that is used for step-up dosing is on its own quite effective. The good thing is that by using the step-up dosing, we can then go to 60, we can go to 90 within a week, 2 weeks, or 4 weeks. So that's relatively soon after they receive the priming dose. So we're not sort of waiting for months before the patient gets the target dose. Okay. We are waiting for just maximum of 4 weeks before they get the target dose. And we've seen already with a pancreatic cancer patient received the 2 priming doses at 30 and then received 60. And that patient's tumor started to shrink and continues to shrink and now is a confirmed partial responder. And this is encouraging because in that pancreatic cancer patient, the tumor had now spread to the liver. And that is by far amongst tumors that have metastasized or spread. The worst place for it to spread is the liver. And we are seeing that we are shrinking the tumor in the liver. So that's going to show that the 30-60 approach that we are using is certainly able to tackle a very challenging pancreatic cancer. So we're really happy with that.
Perfect. All right. The next question, in a prior earnings call, you stated that cohort 6 may not be necessary and is to be determined. What are the reasons? You're already seeing what you wanted to see, or there are safety issues that may be problematic at higher doses than 90 micrograms per kilogram?
Yeah, so, when we look at the data, when we compare data at 10, 30, 60, we've consistently shown that as we're increasing the dose, the population of lymphocytes, the CD8 T cells, the cancer-fighting immune cell population, that's increasing with the dose. So we're saying, okay, if it's increasing, why don't we push it to even higher levels, to 90 and perhaps even 120? The thing is that as long as it's trending higher, and as long as the drug is safe, it's best to give the patient the best opportunity to respond to treatment and have a higher percentage of patients having CRs and PRs and stable disease. So it is to, I would say, for the patient's benefit, that as long as the drug is safe, the higher the dose we can proceed with, the better it is. Now, if on the other hand, we see that there is no further increase at 90 or 120, and we see that this is actually, we've peaked out, we've maxed out the immune system, we can't proceed any higher, then obviously the need for cohort 6 goes away. Okay. So we'll play it by ear. We don't make that ultimate decision. We listen to the KOLs, the doctors that are treating the patients, and get their feedback and input and make sure that we are on the right track. We don't want to be in a situation like Sanofi, where they came back and realized, oh shucks, our dosing was not optimal. We have to go back to the drawing board. And this is after treating 150, 200 patients. We don't have the luxury of saying, we'll just go ahead at 60 micrograms or 90 micrograms. And we don't have the luxury of going back to square one and losing 2 years and realizing we didn't do enough. So we want to avoid that. So we'll do the best we can and get as high a dose and the most robust dosing regimen for the patients, as long as it's, yeah.
No, that makes perfect sense to me. And I mean, I'm happy to see that so far there haven't been any dose limiting toxicity. So, and it looks like the PK data has been just really solid. No, like any adverse effects it looks like they were very mild, and
you'd expect from the mechanism, you'd expect these, and you can see 92% of the patients are just grade 1 or grade 2 events. Most of them went away within the first 24 hours, at the most 2 days. So very transient. And because we're using step-up dosing, we find that, in fact, the incidence of those adverse events actually diminish substantially, so that your second, third, fourth dose actually is even more safer and therefore more better tolerated. So I think most of the adverse events you see that we've listed are mostly in patients when they get the first or second dose. But subsequently, those adverse events disappear.
Yeah, I'd rather see them on the front end,
yes,
and see them as the drug accumulates.
Correct.
All right. Do you know which tumor types the MDNA 11 and checkpoint inhibitor dose expansion trial will focus on?
Yeah, good question again. And this is something we have previously planned, as our protocol would say, that we would enroll patients with melanoma, renal cell carcinoma, and potentially other tumor type. Now, this is still in the works. We certainly will be working closely with our KOLs We will be collecting data that we've been seeing, for instance, patients with sarcomas, with pancreatic cancer, for instance, seeing a response there. If we keep on seeing those trends, we might decide to make some additional changes to the protocol. It's not finalized. And finally, with respect to combination part, we would have to work together closely as to where the unmet needs are, where checkpoint inhibitors do not do the best job, which is the indication that we can add MDNA 11 to and make the checkpoint inhibitor work even better. But those are the things we'll be discussing with our partner, Merck, in terms of making those final decisions. So we're not disclosing any of that at this time. Okay.
Okay. Fair enough. What is the IP status of MDNA 11? Stanford has a patent to cover Super 2. Does the company have patents to further cover non-alpha, IL-2 albumin fusion, and IL-2 FC fusion for BiSKIT program?
Yeah, so a number of things that, of course, Super 2 is something we've in-licensed from Stanford, and we own exclusive worldwide rights to the Super 2 platform on its own. This is from Stanford. We have subsequently added some additional IP around creating the long-acting version. We've added new mutations into the Super 2 molecule itself, the fusion with FC, the fusion with albumin. Those are sort of new IPs that we've included, especially the BiSKIT, where we have fused our molecule to a checkpoint inhibitor. So that is completely brand new IP, and that sort of stretches our patent portfolio all the way into 2038, 2040. So gives us extensive runway in terms of IP protection. Okay, so those are the new IP that we've filed.
All right, so I heard exclusive worldwide rights.
Yes.
Those are
that is exclusive worldwide rights. Yeah.
Okay. There were a lot of other words there that were pretty big for me. So
okay,
after promising MDNA 11 data, are you willing to sell the company at the right price?
Well, it's very, there's, if you look at Medicenna, we have MDNA 11. That's really exciting. It's something that, Sanofi, based on Synthorx's early data, just on their IL-2 program, made a huge offer and purchased it for $2.5 billion, right? So now remember, Synthorx had a number of other earlier stage assets. So, if it's being driven by an IL-2 program, and if we see that continues to share data on not only MDNA 11, but our BiSKITs program, it is very likely that on the back of some promising single agent data, this is really important to sort of, the bar has been raised as far as any acquisitions that take place. People want to see MDNA 11 show a handful of patients responding to therapy. And I think if that happens, it'll be very likely that we have a suitor out there and make an offer. And certainly whatever is ideal, necessary, best for the shareholders, will do that. As you know, I personally own a big chunk of the Medicenna shares. So whatever I do will be for the best of all the shareholders. So we'll see. I mean, it's early to say, but we also have opportunities to just sell the asset, the molecule, the technology, just the Super 2 platform. Remember, we have Super 4 and Super 13 as well. And those could be separate programs that a pharma company may not be interested in for now. So we'll look at different opportunities, whatever is best for the company, whatever is best for the patients. In the end, the drug has to be in the marketplace. We know that a drug such as this needs a big partner so that the Keytruda, the Opdivo sales that we have seen of multi-billion dollars every quarter, I think we are north of 20 billion now with these checkpoint inhibitors. The opportunity is there, and I think that's something that Medicenna cannot take it all the way, and therefore the sooner we're able to partner, the better. Okay.
This is one thing that, as a shareholder, I get a lot of comfort being able to buy at a $30 million market cap when I know that the data has been good.
Yes.
this is a fast process. We're not sitting around waiting a decade to get news here. And that, at 30 million, knowing that acquisitions are taking place in the billions,
Right.
This feels like a really, that the risk reward here is pretty insane. So I'm happy with it.
Yes.
So
No, I agree. And I think we know that pharma, we've seen that with Sanofi, we've seen that with Roche. They continue to invest in the IL-2 programs. Despite the challenges they've seen, they know that they have to go back to the drawing board and work at it. They believe in IL-2 as we do. And I think our data continues to show the potential promise there. So we think we're on the right track, and we look forward to sharing more data next year. So really excited about middle of next year, especially. Okay.
so am I. All right. So the next set of questions are on BiSKITs. So
Right.
we go. What are the primary competitive advantages of MDNA 223 anti-PD IL-2 BiSKIT? Do you have any concerns about toxicity of this molecule?
Right. And this molecule is very unique in the sense that it has two functionalities built into one molecule. That means down the road, if you look at our data, now, of course, these are all mice data, but when we treat these mice with our IL-2 MDNA 11 or MDNA 19 on its own and combine it with a checkpoint inhibitor, both of those scenarios really slow the tumor. But when you create this BiSKIT, where you sort of linked our IL-2 with the checkpoint inhibitor, it's sort of 2-in-1 molecule. That BiSKIT has even better efficacy than the two different checkpoints and MDNA 11 injected separately. So what it does is the key advantage is you treat and you use just one drug to achieve your objective, right? So that's one thing. Second is that you're able to stimulate the cancer-fighting immune cell simultaneously. The same immune cells that gets the checkpoint inhibitor also gets the IL-2 at the same time. So that makes it even better. And so those are the two things. The question is, well, what about the toxicity issues? And that's where our step-up dosing really helps. So that's what we've also solved for the BiSKIT program. We've sort of been able to file a patent around our IL-2, our step-up dosing strategy. It doesn't only apply to MDNA 11, it applies to our BiSKITs as well. So ultimately what happens is that even if you're dealing with toxicity issues, of course, it's a lot more potent. The thing is that by using step-up dosing, we can reach the higher doses safely. So that's important.
Well, that leads perfectly into the next question. So why does your anti-PD-1 IL-2 BiSKIT and MDNA 413 BiSKIT utilize MDNA 19 rather than MDNA 11 for the molecule's IL-2 component?
Okay, so let me sort of differentiate between MDNA 11 and MDNA 19. Okay, the only difference between MDNA 11 is that MDNA 11 has albumin to make it bigger and stay in the bloodstream longer. MDNA 19, on the other hand, has a different protein scaffold. We call it FC. Again, it's present in our bloodstream. It's sort of an antibody. We call them IgG antibodies that are present in large amounts in our bloodstream as well. So we've used these two interchangeably. It's just that most antibodies, like the checkpoint inhibitor, those antibodies, the scaffold they have, the protein structure they have are very similar to FC, okay? And therefore, to minimize too much complication in the project, in designing the molecule, what we do is instead of when using the anti-PD-1 and our IL-2, you already have the FC portion in anti-PD-1 anyways. So you might as well take advantage of that and not make the molecule more complex. So it's a simple thing to do. Take the anti-PD-1, which is 95% the FC molecule is there, and you mix it or link it to our IL-2 and bingo, that's what happens. Now, the question about 413, right? So 413 is, it's again interchangeable. We can flip and use either the FC or albumin. We are testing both of them just like we did with MDNA 11 and MDNA 19. We took it all the way to monkey studies. Once we found which one of those was better, we stayed with that. Same will happen with MDNA 413. If we find that the albumin version of 413 is better than the FC version of 413, we'll stay with albumin or FC, whichever is the best in that situation. So yeah, we haven't finalized 413 as whether it's FC or albumin. We'll decide that once we have some monkey data.
Okay, perfect. What are the potential competitive advantages of MDNA 413 compared to other prominent IL-4, IL-13 drugs like Dupixent?
Yes.
Okay.
So Dupixent is being used for treating patients with atopic dermatitis, different respiratory diseases like asthma. It's already a $10 billion drug for Sanofi. So it's a huge market opportunity. The thing is that Dupixent is a large protein. It's quite large. It's an antibody. Okay. Our drug, we call 413, we can make it large by mixing it or combining with albumin, or we can not make it large. We don't attach albumin or FC. So it makes the molecule small. And why is that important? Because if you have respiratory diseases, what is the best way to treat a patient? Is to nasally deliver the drug by a nasal spray. That's it. Whereas Dupixent, you have to actually go and get injected on a regular basis every 2 weeks. And to treat patients with asthma or atopic dermatitis, which we think our drug would be able to apply on the skin, it gives a lot more ease of use for the drug for patients who do not want to go and get injected every 2 weeks or every month. Here, this is something that can be administered intranasally very simply. So that's one big advantage, okay? The second advantage is the design of the molecule. The antibody that is used for Dupixent, that particular antibody binds two types of receptors. It binds to something called the type 1 IL-4 receptor and the type 2 IL-4 receptor. For the Dupixent molecule to work, actually, it should only bind to type 2. You don't need it to bind to type 1. So there is potential for side effects and other issues with binding to type 1 receptors. Our molecule is designed so it's even more targeted, even more specific. It will only bind to type 2, not type 1. So there is a therapeutic benefit, there's a safety benefit, there's ease of administration, and therefore potentially a much better version of Dupixent that we have. So that's obviously, we are focusing first and foremost in the oncology side, as quicker for it to get into the patient, into humans, to be able to be tested. And once you have that established, you can then transition and look at the same molecule or a sort of adapted version of 413 for other indications. So that's a big difference.
So when you're talking about other indications, did you say intranasal?
Yeah, intranasal.
So
right.
So you're not trying to get into alveolar sacs.
Yeah, so
like the mucosal membrane.
Yeah, you could basically use a spray, and you have the ability to use a nebulizer. You can use a spray, depending. Sometimes you have inflammatory conditions that occur in the upper respiratory tract. That can be treated with MDNA 413, or you might have patients that have asthma, and therefore they need a nebulizer, just like they use today to they use steroids to reduce inflammation, and they use nebulizers. You can use the same kind of nebulizer to deliver 413 into the lungs, in the alveolus, and therefore treat patients with asthma and so on.
Okay.
So that's the opportunity that you can't do with Dupixent. It's too large. You can't it's a huge molecule. It's about 10 times bigger than our 413.
Wow. Okay. So not just a little bit bigger.
10 times bigger, right.
All right. Well, my final technical question is really just about partnership for MDNA 55. Given the fantastic phase 2 results and FDA meeting, what are the issues with closing a partner? Valuation, others? What is plan B for this program?
Yes, so it has taken us a while to, it's been 2 years since we met with the FDA. Since then, we've had a number of discussions with different companies. The key aspects that were sort of not addressed was, would neurosurgeons actually use our drug? That was the important thing. Remember, it's administered directly in the tumor. So that was one question. They wanted to know if insurance companies would reimburse. And if they were, what is the pricing going to be for MDNA 55? So those were the big commercial issues that needed to be sorted out. So we went back and did a lot of work with different consulting firms and interviewed about 40 different key opinion leaders and talked to 10 insurance companies and interviewed them. So that gave us a much better feel for what the pricing will be, what the uptake will be, what will neurosurgeons do with this drug. So we've collected all that data, and we are at a point now where we've been talking to multiple companies. That discussions are still underway, but the challenge is glioblastoma is such a difficult place to be. There's been so many failures with GBM. They've always seen, the big pharma is saying, oh, great data, phase 2 looks fantastic. And then suddenly it fails in a phase 3 trial. So not surprisingly, the companies that are currently evaluating MDNA 55, they're spending a lot more time in the data room. They're going through each patient individually and seeing, what treatment they received, how they did. So they want to look at all the safety issues. So they're spending a lot of time in the data room. And that, in a sense, is good because it shows that they're interested in the program. So we will announce it when it happens, but we just have to let that process to run through.
All right. Well, that's another good thing for new shareholders to be aware of, that there already is a saleable item out there. So that's a good point. Well, thank you both for joining me today and answering all of the questions that were sent in. And I really look forward to next year.
Yes, well, have a great holiday season and look forward to catching up again. And hopefully, as I said, multiple opportunities to share new data. Q1 next year, middle of next year with monotherapy, and then the end of the next year for the combo with Merck's Keytruda. And then, of course, there could be a surprise component with the MDNA 55 partnering. So we'll leave it all there. Okay.
Excellent. Wonderful. Thank you.
All right. Take care. Bye-bye.